Human beings, dogs, oak trees, and tarantulas, they all reside on the complex end of the microorganism spectrum, whereas bacteria look pretty simple, right? Well, it turns out that even single-celled bacteria exhibit their own astonishing complexity. Just how simple can a cell be and still exist? Our guest today examines that question and how it affects our understanding of where we came from. Coming up on today's edition of Origins, Minimal Complexity Mystery with Dr. Paul Nelson. Hello and welcome to Origins. I'm Ray Heiple. It's an honor to be your host today. During this program, we showcase interesting guests who present evidence from science, along with other important facts validating the truth of creation and the accuracy of the Bible. Our guest today, Dr. Paul Nelson, studied evolutionary theory and the philosophy of science at the University of Chicago. He's currently a senior fellow of the Discovery Institute and adjunct professor in science and religion at Biola University. Paul has been involved in the intelligent design debate internationally for more than 2 decades. Welcome to the program, Paul.
It's great to be here.
Minimal complexity mystery. Well, if it's a minimal complexity, I think I can understand it.
Well, it turns out actually to have all kinds of interesting implications, both for the origin of life and how we think about biology. And the question we're asking is really quite simple. It's this: how simple, pun intended, can an organism be and yet exist? So we're not talking about blue whales or oak trees or T-Rex or anything big. We're talking about the simplest conceivable free-living organism, which would be a bacterial cell. What does that thing need just to survive? So that's the question, and we're going to look at how that question is answered and what implications those answers have. So let me go to the board and show you the theater where our drama will take place. Here it is, and we have 2 major zones, if you will. Down here, you're not alive, all right? So those are stones in your garden, the asphalt in the parking lot, unquestionably non-living. Up here, you are alive, right? So these would be you and me, or the bacteria in our large intestine, anything that's respiring and metabolizing and reproducing and performing the functions of all living things. Now in here we've got an interesting kind of gray zone. For instance, you could have a complex virus here, right, that's got lots of DNA, hundreds of thousands of base pairs of DNA. But it turns out that to replicate that DNA, the virus needs to borrow equipment, if you will, from a bacterial cell present up here. So we could say, well, it looks pretty complicated, it's got a lot of DNA, but if it didn't have a cell nearby, it wouldn't exist at all. So we've got this interesting gray zone, put viruses in there, but 2 places or 2 regions where you're definitely dead, not alive, and you definitely are alive. Now, we're going to try to figure out, using a naturalistic framework at the start, how do these zones relate to each other? So the question for minimal complexity is this. We know we have cells, and according to evolution, they started down here. Now, the usual approach to answering that question of how those cells came to be is to go what's called bottom up. And there's a reason for that. Most evolutionary theorists look at this and they say, well, once upon a time on this planet, there were no cells, just non-living chemicals, and by some unknown pathway, cells came to be. So let's recapitulate or try to recreate, they say, that pathway, whatever it happened to be, starting with things that aren't alive and trying to achieve the living state. You can think about this like looking for the sufficient conditions for the living state, or more simply, the recipe for life. Okay, so if I gave you flour and eggs and the right kind of container and sugar and some other things and heat, you might be able to get, or you should be able to get, if I give you the right recipe, a cake or a muffin out of that. In a biological context, what we're looking for are are the sufficient conditions or the recipe that will give us life. So starting bottom up, we're going to put together all the components needed to give us a living cell. Let's say we've got 12 of them. Doubtless there'll be more, but let's just think about 12. We put all these together and boom, life arises. Okay? Now that's the usual approach in a naturalistic evolutionary framework. But we can come at the problem from another direction, not just bottom up, but top down. In the top down approach, we start with cells and we subtract. We take things away and we say, hey, did you need that part? Think of me, for instance, right? Here's my right arm. I like my right arm. I'm right-handed. But we can imagine, kind of grisly thought experiment, going into a surgical suite, taking my right arm off. I come out of anesthetic. I'm unquestionably alive, very unhappy, but I can carry about most of my business. What can't I do in that same scenario?
Well, they couldn't take your heart out.
They couldn't take my heart unless they replaced it with something. Mm-hmm. They couldn't go through my brainstem. Mm-hmm. If the surgeon took a scalpel and disconnected my respiratory control centers back here where they are, it's over for me. Mm-hmm. So not all the parts of our biology are equally important. Mm-hmm. So this approach looks for the necessary conditions for the living state. You start with life and you take things away. And you say, are you still viable? Can I still grow you in a test tube? Now you're looking for the parts list for life, where you say, all right, I'll start with a cell, and I'll take things away and ask the cell, are you still able to make a go of it? So here we've got our same 12 parts, and I say, well, I can do without F, cell's still there. I can do without H, yeah, still growing, still alive in that test tube. I take away J, and I lose the whole system. It turns out then that J is an essential component. I can't do without it. So by this kind of subtraction analysis, we can work top down and say, where's this lower threshold? How much complexity do I need? Now it's very important, we'll come back to this later, that these two arrows meet in the middle. If they don't, the naturalistic approach to the origin of life is going to have real problems. Now, let me give you the main bottom-up approach, current in evolutionary theory, for how this happened by an undirected chemical pathway. It's called the RNA world hypothesis. And it's thought to solve a classic chicken and egg problem related to the origin of life. You know what a chicken and egg problem is.
Sure. Which one came first?
Which one came first, exactly. So let's start with DNA, which gives us the instructions for building proteins. So DNA is a wonderful repository for information, very stable, storing the specifications that will give you proteins. Proteins do most of the work in cells, transport, metabolism, signaling, all that sort of thing. But there's a problem. The information needed to build proteins is stored in DNA. But to get that information out of DNA, you need proteins. When you look, for instance, at how DNA is translated into protein, it's a protein story. Proteins are active all along the way. So origin of life researchers looked at this puzzle and they said, to have proteins, I need DNA. To have DNA, I need proteins. It looks like I'm locked in a closed loop here. How does RNA solve the problem? Well, here's how the hypothesis works. RNA, like DNA, is a nucleic acid. It's a long linear molecule that can store information. So it can perform that information storage function essential to all cells. Unlike DNA, however, single-stranded RNA can fold up into interesting shapes. I've got one illustrated here, and do jobs for you in the cell, very much like proteins. So it seems to solve this dilemma. You've got one molecule that can both be an information storage medium and an actor.
So it can play both roles.
It can play both roles.
Okay.
So the hypothesis is, coming bottom up, the first living things weren't cells. They were self-replicating RNA molecules called ribozymes, that's their name, and somehow they come to be, and this arrow of complexity increases, and eventually you get the cellular state. Okay. That's the standard theory.
Sounds reasonable.
Yes, but there are problems. So Stuart Kaufman, who works on the origin of life at the Santa Fe Institute, said, it's an elegant, interesting hypothesis, but I don't buy it. And here's his reason. he said there's a problem with this view. He calls it the nude replicating RNA molecules view. And he says the problem that's most insurmountable is the one that's least talked about. All living things seem to have a minimal complexity below which it is impossible to go. Now, that's a really interesting statement to make. Why would he say that? Well, the answer comes from cell biology itself. In fact, I've got a video clip here that will help us understand why there is this minimal complexity for any kind of cell that we can imagine. What it shows is what any living thing has to do. There are certain basic functions that all life, from you and me right on down to single bacterial cells, all life has to do these basic functions. So let's take a look at that.
If you're going to be the first living cell, you need a boundary, a membrane that protects you from the rest of the environment. This membrane must selectively allow the passage of specific nutrients and raw materials required for fuel and energy. You'll also store huge quantities of genetic information in coiled strands of DNA. These detailed instructions for life are copied with remarkable precision. Then, in factories called ribosomes, this information is converted into chains of amino acids and proteins. Finally, you'll be equipped with everything you need to reproduce future generations of cells.
All life does this. So you and I have a boundary between ourselves and the environment. We don't let just anything in, we don't put just anything out. We take in raw materials and convert them into things that we can use. We replicate, although not like bacterial cells. We have more interesting ways of passing on our genetic information. But all living things have to do this, and all living things have a minimal complexity threshold. As I said a moment ago, I can't do without my brainstem, although I could do without my right arm for most of the basic functions of my biology. So Kaufman looks at this RNA world scenario and he says, every cell that I know, there's a bottom line down here below which you can't go or you're dead. Now, what does that mean? Well, it means that the RNA world hypothesis has got to come to grips with the reality of minimal complexity for bacterial cells. That's what we're looking at. And Kaufman says, look, everything that we know has at least the diversity of Pleuramona. Now, that's a fancy name for a kind of cell called Mycoplasma. Mycoplasma normally infects the epithelia of the human airway. We find it there. We also find it in the human genetic tract. And it's a very stripped down cell. You can't grow Mycoplasma unless you give it very, very rich culture media. Like it needs to have all its amino acids supplied. It needs cholesterol. It needs all kinds of raw materials that it can't make for itself because it looks like it's jettisoned most of that hardware. So this is a very stripped down cell, has only slightly more than 500,000 base pairs of DNA, but it is a free living cell, so it provides a pretty good model for what an early cell would look like.
Okay, so this is some primitive, a very primitive cell in terms of not a lot of complexity, not a lot of parts, relatively speaking, compared to other cells, and yet it's alive, it can replicate.
It's alive and it can replicate. So it does provide us a good model of what a simple cell would be like. Now the problem is, Kaufman goes on, this minimal complexity won't go away. Why is it there? Well, it's there because when we do this top-down analysis and take parts away, we discover that we can't take away very many and have a functional cell. So we can do this actually in reality as an experiment with Mycoplasma, and this turns out to be its parts list.
Are you saying that one cell, that simple cell, that primitive cell, has all those parts?
And actually more. They're not all listed here. But some of these parts are enormous in number. For instance, this group of parts, all under the heading of translation, are involved in the machine in the cell called the ribosome that builds proteins. Here's an easier way to visualize it. We can take those parts and put them into major functional classes. So within each of these functions, there will be lots of proteins. The one I just pointed to here, DNA translation, there's dozens and dozens there. Cellular processes like cell division, dividing yourself as you're reproducing, that's going to have lots of proteins. So each of these subfunctions will have a whole bunch of separate parts within them. Let's look at some of them.
And one cell needs to do all those functions.
One cell needs to do all of them.
Okay.
So this protein is involved when cells divide. They need to find their midline, their 50% point, so to speak, because that's where they're going to divide, putting a whole chromosome in each new cell. And this protein helps to do that job. It locates the midline. Now, think about this shape and compare that shape to this one. In fact, this is a multi-protein complex. It's a big machine, if you will, that functions as a nursery for baby proteins. Many proteins, as they're formed, are very sticky, and in this central chamber here, the chaperone, as its name implies, provides a space for the proteins to fold so they can be released later to perform their function. If you don't have chaperones in your cells that are making proteins, you'll die, because you need this to enable those proteins to fold properly. Okay, keep that shape in mind and compare that shape to this one. This is yet another essential protein evolved in the actual machine, the ribosome, that builds proteins. And you'll notice its shape is different because it's doing a different job. So we could work our way through that whole parts list, looking at those different molecular actors and say, do we have to have all of them? Now, this experiment has been done several times, in fact, and the results are always interesting.
Let's hold it right there, Paul. We're going to take a break. We're going to see those amazing results and what they mean right after this. Stay with us. Welcome back to Origins. We're talking to Dr. Paul Nelson, who's been sharing some fascinating things about minimal complexity mystery that looks at what are the minimum amount of parts necessary for an organism to be alive. Where do we go from here?
All right. When we last spoke, we had the question, how much can we take away and have a functioning cell? Which to me is just fascinating because it says there's some lower discoverable threshold below which life ceases to be. Now, that experiment has been done several times, in fact, and the results are always fascinating. The first time it was done was in 1999 by a team led by Clyde Hutchison, and what they found was quite remarkable. Now, what they did was to use a piece of DNA called a transposon. Transposons integrate themselves randomly into other DNA, and as they do, they disrupt the DNA where they land, all right? So, thought experiment. Let's say I give you a Winchester pump, right? Shotgun, 12 gauge, and a big box of shells, all right? Double-aught buck, right?
Okay, I'm going to be happy for the rest of the day.
Okay, and we're going to go to your personal library, all right?
Oh, okay. No,
all right,
I don't like
your
the way
theology
this is
library,
going.
all the
No.
books are there on the shelf. I blindfold you. We fill up the magazine of that pump Winchester, and I say, just keep shooting. And you randomly blast away.
Can it be my assistant pastor's library?
All right, we'll take his library. You randomly blast away, and then we take off the blindfold, we walk over, we see what books survived. All right, this piece of DNA does that. It integrates itself blindly into other DNA, and as it does, it disrupts the genes where it hits. So here's our transposon, right?
Okay.
And it's going to come along, and it's going to land right in the middle of that piece of DNA. Now, that piece of DNA is coding for a protein the cell cannot do without. And we know that because when this happens, the cells die.
So when that hits, it dies?
What happens is it integrates in, and when the cell needs to produce that protein for some role in the cell, for some function, it's not there, and the cells die at that point.
Okay.
And we know that because we try to get them to grow, and there's no activity at all. They're stone dead.
So the experiments have proven this.
Right. What they do is they put the transposons into the cells, and here you can see you've got 4 integration sites. At least one of those little red hatch marks hit an essential gene. No activity, no growth. Cells are dead. Same thing happens here. At least one of those three hit something the cell couldn't do without, no activity. Now, sometimes, however, the transposon goes into the cell and integrates, and the cells are alive.
So it has to hit in just the right place.
It has to hit in just the right place. But the very fact that you see transposons here and here and here in the DNA of a living cell tells you these sites were probably not essential. So think about your car. Its main function is to get you from A to B, right? So I can go out there in the parking lot and I can remove, let's say, the DVD player. I can probably take out the radio, you'll be fine. I can't take out the fuel injector, right? I can't take out either of the axles. All right, so in a sense, you could think that's the radio, that's the DVD player. For the basic functions of your car, those parts can be dispensed with. Now, from that information, these scientists discovered just how complex even the simplest known cell has to be. Here's what they found. It's really amazing. Of the 482 protein coding genes for that bacterial species, over a third were essential under laboratory growth conditions, including 110 genes of unknown function.
So they could remove maybe 100, but after that, they couldn't remove any more.
Then you hit that bottom threshold. You hit that basement layer below which you can't go and remain alive. Now, the real kicker for this result is that 110 genes of unknown function. Nearly a third of the essential hardware of the simplest cell that we don't know what it's doing. And we don't know where it came from. In other words, these genes have no known evolutionary relatives.
We can identify them as genes, but we don't know why they're there.
We don't know why they're there, but we know that cell can't do without them. That's a remarkable finding. That's the kind of thing that gave me goosebumps. Because if we go back to our, well, here's what they said in the paper. It's remarkable. The presence of so many genes of unknown function among the essential genes of the simplest known cell suggests that all the basic molecular mechanisms underlying life may not yet have been described. That's exciting because that says we've got mysteries here about very, very simple cells that have yet to be solved. But here's the evolutionary puzzle. We've got this complexity below which it is impossible to go. If that dotted line is somewhere in the neighborhood, let's say, of 350 to 380 proteins, that's going to be a hard target to hit for an undirected chemical pathway, as naturalistic evolution postulates.
So it needs 350 to 380 accidents.
Major accidents.
To happen exactly the right way, at exactly the right time, and in exactly the right sequence.
Here's an easy way to think about this puzzle with relation to something that human beings have a lot of experience with, and that is tunneling. All right, so let's say we've got a mountain in Switzerland, or maybe even here in Pennsylvania. We want to put a tunnel through it, and we've got two crews. One working from the east, the other working from the west. Okay, so here's our east crew, crew 1, they're tunneling in that way. Here's crew 2 coming from the west. Now, it's intuitively clear that these two crews have got to be in communication with each other. That's a big mountain, right? And unless they know where they're going and where they're going to meet, they're going to miss each other by a mile. They want to meet somewhere here. But already you can see in this diagram, they're going to miss.
Somebody's going to be fired on this one, I think.
Yeah, someone's going to pay for the mistake. So if we apply this metaphor to our problem, the bottom-up approach to the origin of life and the top-down approach have got to meet in the middle. If it turns out, however, that top-down analysis says 350, 300 proteins, that's your bare minimum, and the bottom-up approach says I can't even get you a single protein, the origin of life scenario that naturalism and materialism offers us is bound to fail. So unless those tunneling crews can meet, well, I got to move my hands like this, but you understand the idea. These two modes of analyzing the origin of life need to intersect in the middle. They need to meet. If they don't, life on this planet did not come to be by an undirected natural process.
Sounds like the evolutionists have a pretty serious problem.
They have a serious problem, and here's the real striking thing for me. The problem doesn't come from the evidence, because the evidence clearly indicates that design was needed for the first cell. The problem is given to them by their underlying philosophy of materialism, which says if you're going to explain any puzzle in nature, you're only allowed to use natural laws and chance. That is too limited a toolkit for the reality of minimal complexity.
They don't want to go where the evidence leads them.
Well, you know what? It's human nature. All of us, I think, if we're being honest, want the world to be a certain way. But I think in science, as in daily life, we need to let truth guide us. And truth is a reliable guide. And if we listen to nature, if we listen to what Solomon calls wisdom, that will guide us into knowledge.
And you shall know the truth, and the truth will set you free.
Amen.
Thanks for joining us, Paul.
It's been a treat.
We really appreciate it. And we appreciate you joining us as well. Today we looked at minimal complexity mystery. What is absolutely necessary for an organism to be alive? Evolutionary theory says that by incremental changes an organism can reach life. But the evidence shows we need a creator to create life. And it just goes to show you that we know what the Bible says is true, and the proof is all around you. Thanks for joining us for this edition of Origins. We'll see you next time.
Thank you for watching this edition of Origins. For a DVD of this series, you can order online or send a $12 donation to cover shipping and handling, and write to Origins Program, number 1903, Cornerstone Network, Wall, Pennsylvania, 15148.
This presentation was made possible by the faithful prayers and financial support of you, our Cornerstone family.